Today we will learn about journal physiology. In the journal physiology, first we will do the functions of plasma membrane. First we will learn about the functions of plasma membrane. So it is a protective membrane that encloses cell body, and it is also called cell membrane and plasma lemma. So the proper functions of plasma membrane: firstly, there is protective function. The cell membrane protects the cell organelles like mitochondria and vacuoles, lysosomes, endoplasmic reticulum, nucleus, and the cytoplasm from external substances by forming a mechanical barrier. So the next function is permeability. Since the plasma membrane is selectively permeable to some substances and impermeable to other substances. And the next function is exchange of gases. Oxygen and carbon dioxide can diffuse into and out of the cell through cell membrane as they are soluble in cell membrane. The next function of cell membrane is absorption. Some substances are absorbed through cell membrane, especially nutrients. The next function is excretion. Some unwanted excretory wastes are thrown out of the cell through plasma membrane. And the next function is maintenance of shape and size of cell. The cell membrane prevents water accumulation and maintains cell size through sodium potassium ATPase channels. Sodium potassium ATPase channels. So the functions of cell membrane: firstly, there is protective function, then permeability and exchange of gases, absorption, excretion, and maintenance of shape and size of cell. Now our next question is, which organelle is called suicide bag of cell, and what type of enzyme does it contain, and what is its function? So lysosome is known as the suicidal bag of the cell. These are membrane bound vesicles within the cytoplasm of cell. Lysosomes are membrane bound vesicles within the cytoplasm of cell. Lysosomes contain enzymes like amylases. Amylases digest carbohydrates, and proteases, as suggested by the name, proteases digest proteins, and lipases digest lipids. Nucleases digest nucleic acid. Now, what is the function of lysosome? Degradation of macromolecules that have been engulfed by the phagocytosis and pinocytosis or endocytosis. So degradation of macromolecules like carbohydrates, proteins, lipids, nucleic acid. And degradation of damaged organelles is also carried out by lysosomes by formation of autophagolysosome. Degradation of damaged organelles occur with the help of formation of autophagolysosomes. Removal of excretory products is also a function of lysosomes, and some lysosomes have secretory functions also. Like example, perforin and granzymes, which destroy microbes, and serotonin by mast cells, and serotonin by mast cells that carry out allergic response, and then melanin secretion from melanocyte. Now our next question is, what are the peculiarities of mitochondrial DNA? Mitochondria, first of all, is powerhouse of cell, and it has an outer membrane which encloses the cytosol, and then the inner membrane is known as cristae, which is thrown into numerous folds and have 5 enzymes required for Krebs cycle. Now mitochondrial DNA. Matrix of mitochondria have DNA, and most of DNA is packed within the nucleus, but a small portion is enclosed within the mitochondria. Small portion of DNA is enclosed within the mitochondria, and the mass of mitochondrial DNA is 16,500 daltons, and the mitochondrial DNA is inherited from mother, as only nucleus of sperm takes part in zygote formation, and other cell organelles are derived from the egg. The mitochondrial DNA is responsible for production of enzymes that take part in respiratory cycle and specific for cells having mitochondria. The enzymes are specific for cell which have mitochondria. Due to small size and easy to isolate, uh, mitochondrial DNA is a target for genome sequencing projects. Mitochondrial DNA lacks introns as against the DNA which is present in the nucleus. And mitochondrial DNA have ability of translation using a single tRNA sequence. Ability of translation. Now our next question is, explain different feedback mechanisms operating to maintain homeostasis with example. First of all, what is homeostasis? Phenomena of maintenance of constant internal environment. Feedback mechanisms. Any change in pattern of any system activates sensor. These signals reach a control center. The effectors bring about necessary change by causing a required action. Firstly, there is negative feedback mechanism. Negative feedback mechanism, a change in homeostasis causes to inhibit the natural activity to bring the change under control. Change in homeostasis to bring the change, natural activity, inhibit the natural activity to control. Example is thyroid hormone regulation. Increase in the serum thyroxine levels, which is detected by the pituitary gland, and negative feedback on thyroid stimulating hormone secretion, which decrease the thyroid stimulating hormone secretion, and thus the thyroid hormone levels like thyroxine levels decreases. Another example of negative feedback mechanism is follicle stimulating hormone regulation and water regulation. Whenever there is decreased in water content, osmoreceptors in hypothalamus stimulates and thus increase the ADH secretion from posterior pituitary, and water retention by kidneys increased. Thus the water content come back to normal. If there is increase in water content, then no stimulation of osmoreceptor in the hypothalamus, then there will decrease in antidiuretic hormone secretion and decrease in water retention by the kidneys, which bring back to normal water content. And next is positive feedback mechanism. In this type of feedback, there is increase in the activity in the same direction. Examples are blood clotting, labor, milk ejection reflex. So in the milk ejection reflex, baby suckles at the breast and afferents reach the pituitary. Afferents reach the pituitary. Oxytocin releases from the posterior pituitary and milk ejection from the breast, and baby suckles more. Now we have question: what is milieu interior? What are the mechanisms which help in maintaining fluid osmolality? Milieu interior literally means internal environment. Internal environment. And Claude Bernard, a scientist in 19th century, explained that multicellular organisms lived in perfectly organized, coordinated environment which is called milieu interior. It is extracellular fluid which house various types of cells and contains many nutrients, ions, and various other structures required for sustenance of cells. It is further divided as blood and interstitial fluid. Blood and interstitial fluid. The milieu interior is divided in blood and interstitial fluid. The extracellular fluid, which is divided in these blood and interstitial fluid. Now, homeostasis in maintaining fluid osmolarity. ADH mechanism. Hypothalamus plays an important role in detecting osmolarity of blood through osmoreceptors. Osmoreceptors sense a variation, decrease or increase in osmolarity in the blood. If there is increase in blood osmolarity, if there is decrease in blood osmolarity due to water loss or inadequate solutes in blood, due to water loss or inadequate solutes in blood, when the osmolarity increased, thirst increases and osmoreceptors are stimulated. Thirst causes person to drink more water. This causes stimulation of posterior pituitary to secrete antidiuretic hormone. Antidiuretic hormones act on distal convoluted tubule to absorb more water through increased formation of aquaporins, increased formation of aquaporine channels, and increased water reabsorption. This causes dilution of extracellular fluid and again decrease in osmolarity. Now the second example of homeostasis in maintaining fluid osmolarity is RAS mechanism, renin angiotensin aldosterone system mechanism. Renin is a hormone which is secreted by juxtaglomerular cells. It is secreted when there is decrease in sodium concentration, sodium ion concentration in thick ascending limb, and sympathetic stimulation causes renin release, and most of the actions are produced due to formation of angiotensin 2 formation. Action on adrenal cortex. Renin stimulate adrenal cortex to produce aldosterone. Aldosterone is a mineralocorticoid and increases sodium ion retention, and thus osmolarity. Increase osmolarity. Now action on glomerular apparatus. The renin constricts afferent arteriole and decreases the blood flowing into the glomerulus. It constricts the afferent arteriole and decrease blood flowing into the glomerulus, thus decreases glomerular filtration and increases extracellular fluid volume and contraction of glomerular mesangial cells, which decreases surface area of glomerulus and increase in sodium reabsorption from proximal convoluted tubule. Now action on the brain. Renin Angiotensin 2 increases water intake by stimulating thirst center and increased release of ADH from the posterior pituitary. Whenever there is increase in water intake, then also there will be increase in the release of antidiuretic hormone from the posterior pituitary. Now we will learn about the examples of positive feedback mechanisms. Firstly, there is example of blood clotting in the positive feedback mechanism. Increase in the blood vessel and bleeding, formation of prothrombin activator, which leads to conversion of prothrombin and thrombin, and then thrombin help in conversion of fibrinogen into fibrin, which leads to stoppage of bleeding. Then there is another example of positive feedback mechanisms. Labor. Onset of labor, contraction of uterus. After the onset of labor, contraction of uterus occurs, which leads to movement of fetus into cervix, and then dilation of cervix and movement of fetus through the cervix, stimulation of receptors in the cervix, discharge of impulse from the receptors and transmission of impulses to the hypothalamus in the brain, and release of oxytocin from the hypothalamus. This is an example of positive feedback. And milk ejection reflex, as we have studied earlier. Baby suckles at the breast and afferent reaches the pituitary in the brain, and the oxytocin releases and milk ejection from the breast occurs and baby suckles more.